Hello everyone, it's BioDare and I dare you to use it. In this video I'm going to show you how to enter data into BioDare. I start by creating a new experiment. First, I have to name it. Please be more verbose than some lab book code. For example, I include the genotype and conditions in the experiment name. The second bit of information we are asked for is what to expect from the dataset, or why it was done. I'm going to write that I was comparing the clock output of PRR79 double mutant with the Y-type in the constant light. Always press accept to save the changes. We have to specify the species we work with and select a data category, which is a mixture of a technique and a data type. It basically helps us to distinguish between common types like LAC imaging and the more rare ones like, for example, PCR data. That was all the information we have been asked for, and we could create a new experiment. However, we encourage you to provide some extra details. It will take you just a few minutes, and you will benefit from it in the future. But more importantly, it will help others to reuse your datasets. I can describe how the experiment was done, for example, the entrainment details. I can also add some comments. By default, the authorship is set to the current user and its institution. But I did not produce those data as I am an IT guy, so I'm going to remove myself and type the names of real author and PI. I will enter myself as the data curator so I can get the credit for putting it into BioDare. I could also provide the funding details. And finally, some measurements details, the technique, the equipment, and recording parameters, like for example that the images were captured every 2 hours. Now I can create the experiment and I will be ready to import the data. If you don't see the required entries for species or technique, contact us by email and we will add them for you. In the future you'll be able to register new terms yourself. Okay, so I press import data and I can upload the file. Currently supported format is an Excel file with the data table in which one column represents time and the others contains values measured at those times. I can either click here to find the file to upload, or I can drag and drop the file into the browser. Then I click to upload it. We are getting these warnings as we have to describe the content of the file. We need to define which column represents time and label the data traces. There are 3 ways in which I can label columns in BioDare. In the first one, I can click and drag over the column's headers to label the whole block of the data. I select data as the column type and enter the label. Then I can continue with the next block. Normally, we design experiments that has the same number of biological replicates, and we can benefit from it in BioDare. If I click OK and Next, BioDare will move me to the next block, so I only need to enter the new labels. It is a good practice to stick to the same number of biological replicates, as it will make your life easier and the results more consistent. I can also use this little form here to describe the data. I define the data range using the column's letters and enter the label. And again, if I have the same number of biological replicates, BioDare will automatically populate for me the next column range and I only need to type new label. The third way of describing data leverages the existing labels in the file. Very often the file already contains column description, like in this example, where the labeling was done by imaging software, so I can reuse these labels. Or maybe some of you prefer to enter text in Excel instead of using BioDare forms. I need to click the row number and its content will be treated as the labels. As you can see, all the regions has been described. But now I need to do some cleaning, as in this example the first few columns do not contain data. I'm going to select them and mark them as ignored. The last remaining bit is providing parameters for the time column. As you can see, I still have the warning present as BioDare does not know it should treat these values as times. I click on the column, I change its type to time, and I need to select time unit. In this case, it's actually image number, not a time, so I need to provide information about interval between images so BioDare can do the conversion. You may remember from the description that the recording was done every 2 hours. I need to say where the values actually starts, as the files can contain some headers on top. And I'm ready to import. A few seconds later, we can see the time series that have been imported. Now we can apply some transformation that help us to visualize the data, like the trending, normalization, or spread. If you would like to use the transformed data in your figures, you can download them as CVS file. I should probably mention the typical problems of data import. One is caused by gaps in the time column. Having gaps in the data values is allowed, like in this file. It is perfectly normal, as some values could be deleted to remove the spikes in the data, or maybe each trace was recorded at different times. That is fine. But you cannot have gaps in the time column. Another problem is caused by non-numerical content. It typically happens if you enter the wrong row number as a starting point when defining time column properties. Or the equipment may be printing some metadata at the end of the file, so check for it. I hope that I convinced you that the data import to BioDare is easy. Thank you, and I will see you next time.